Welcome to our last and third lecture on Chapter 11, How Genes Are Controlled. In a previous lecture, we looked at how gene expression is regulated and controlled, and then also how plants and animals can be cloned. This lecture is going to focus on sort of the last big idea from this chapter, which is the genetic basis of cancer. So you're sort of looking at a mass of growth here occurring in, it looks like, the colon. So cancer does have a genetic basis. It's not just mutations, but essentially what happens in cancer cells is that their mitotic processes are just all sorts of screwed up, and they just sort of divide uncontrollably. And when that happens, sort of those screw-ups in the cell cycle occur due to sort of screw-ups in the gene expression that affect the cell cycle. So for instance, you can take a proto-oncogene, which is sort of this normal gene that sort of helps regulate the cell division process, and it could potentially mute into something called an oncogene. When you have an oncogene being expressed, the cell sort of just divides and divides and divides and divides, sort of just completely sort of out of control, and you end up with a tumor. The other way that sort of this can happen, and we can sort of screw up the cell cycle, is we have genes in the body that are tumor suppressor genes. They sort of tamp down on growth and sort of tumor development. But if those tumor suppressor genes get inactivated, well, that will also lead to sort of this uncontrolled growth. So this is the sort of oncogene process. So here you have nice proto-oncogene. It codes for a particular protein that helps sort of activate cell division. If you get if you get a mutation within the gene, it can turn into something called an oncogene, and now we'll get sort of this kind of hyperactive growth. If you get sort of multiple copies of it, you'll get sort of excess. And so by sort of turning the proto-oncogene, sort of the normal gene, into sort of this oncogene, we sort of get this excess of this protein that helps cell division, and we have this excess of this sort of cell division kind of activating protein, and so we're going to activate cell division a lot, which leads to sort of this kind of uncontrolled growth. Here is a tumor suppressor gene. If it is activated, we get sort of this growth inhibiting hormone, and cell division works pretty normally. But if the tumor suppressor gene sort of mutates or gets inactivated, then you could end up either with no sort of growth inhibiting protein, or some sort of defective, like completely weirdly shaped, non-functional inhibiting protein. And then the cell division just kind of takes place uncontrollably. So both the proto-oncogene to oncogene mutation and the tumor suppressor gene mutations can result in sort of the uncontrolled growth that is sort of the hallmark of a cancerous cell. Typically what happens is sort of not usually just sort of one mutation, but sometimes there are sort of mutations all along the way. Colon cancer exemplifies that quite well. So here we are looking at sort of the wall of the colon, the large intestine. One of those oncogenes gets activated, and so the cell starts to divide. That then leads to one of those tumor suppressor genes sort of turning off, and so now we're getting even further division. We might get a polyp, a benign tumor, but then you might get a second tumor suppressor gene being turned off, and now you've got this really just crazy unchecked growth, and now you have a malignant tumor. And of course the problem with cancer, or one of the problems with cancer, is that if it metastasizes, meaning sort of escapes from its original place, it can then sort of spread through the body. And so essentially what you're looking at is sort of not just a single mutation, but you have a normal cell that goes through one mutation, which then sort of leads it to another mutation, which is sort of like a snowball going downhill, right? Like the worse it gets, the more it mutates, and the worse it gets until you get this sort of malignant cell that's just growing uncontrollably. So here's a nice little kind of check your understanding question. Epithelial cells, which are the cells that line our body cavities, or like our skin, they are very, very frequently replaced. For instance, you lose about 50,000 skin cells a minute, so we're constantly making new skin cells. Do you think that would lead to epithelial cells more likely to become cancerous sort of more frequently or less frequently than, say, I don't know, a nerve cell that doesn't divide as often? What do you think? If you said epithelial cells would be more likely to develop cancer, then you would be correct. Every time we go through DNA replication is a chance for a mutation to occur. And so epithelial cells, which go through DNA replication a lot because they go through mitosis a lot, have lots and lots of opportunities for mutations to occur. And if mutations occur, sometimes they can lead to cancer. Not always, but sometimes they can lead to the development of cancer. Very often, sort of in our just sort of developmental processes, we use those sort of signal transduction pathways. Again, think sort of relay race. And a lot of these proto-oncogenes and tumor suppressor genes sort of code for proteins that are sort of active in these pathways, kind of regulating cell division. And so by sort of screwing up the proteins by activating or inactivating perhaps some of these genes, we sort of get funky, weird proteins controlling the process, and a process that is not proper. So here, for instance, we're looking at a sort of single transduction pathway. Here on the left, it's normal, right? So here's our little growth factor. That's our little signal molecule. It binds onto the target cell, and we get sort of the we sort of activate a gene, right? Get normal product. We get sort of this relay occurring. We get some transcription factor being activated, and we get some sort of transcript that is then translated into one of our proteins that stimulate cell division. But since it all happened quite normally, then we have nice normal cell division. But here, in our sort of mutant, there is no growth factor that has been produced. The gene for that growth factor did not get activated. And so rather than sort of the normal product of this gene, we get this sort of hyperactive relay protein. It's an oncogene. And so we get sort of lots and lots of this transcription factor being made. And we get sort of lots and lots of this transcription protein being made from translation. And so now our sort of cell division protein that stimulates cell division, we've just got way too much of it. And so we're doing way too much cellular division. Here is with a tumor suppressor gene. So here in our sort of normal pathway, there's sort of this growth inhibiting factor. It's going to bind onto our receptor protein, going to go through those relays, and you sort of get this normal transcription factor that activates a particular gene that results in a protein that inhibits cell division. And so we have sort of no cell division taking place. We have kind of normal growth. But in the mutant, the we get sort of a screw up. We still have our growth inhibiting factor sort of binding onto our receptor protein, but we get sort of this screw up in the relay and sort of passing the message. And so you end up with sort of this oddly shaped transcription factor. Well, this particular transcription factor, since it's not shaped properly, cannot transcribe our DNA. And if transcription can't occur, then the translation is not going to occur, and we're not going to get that sort of growth inhibiting protein, and sort of leads to unchecked cell division because there's nothing there to stop it or slow it. And so again, both with the oncogenes and the tumor suppressor genes, we can sort of end up with cancer in one of both ways. Now, of course, cancer is not completely genetic. There are lifestyle kind of choices that we make. If you like to sun worship, right? Maybe go to the beach in the summer and bask in the sunlight. I like to do that. It's not really very healthy for me. Cancer is the second leading cause of death here in the US after heart disease. That is sort of the leading cause of death. That's true not only of here in the US, but in most of our sort of peer countries, sort of what we think of as first world countries. And a lot of times the mutations that occur are due to environmental factors. So for instance, if you're out in the sun too much and you don't put any sunscreen on, or if you are exposed to other carcinogens. So a carcinogen is essentially any agent that can alter the DNA and lead to the development of cancer. And so if you reduce your sort of exposure to carcinogens, then sometimes you can reduce your risk of developing cancer. So you can see, if we look at some of the data here, what the yellow and blue tell you is sort of whether this particular cancer can be screened for. As we move sort of from right to left, gives us how many sort of diagnoses were occurring. And then on the pathway here, on the y-axis, it's looking at deaths. So this data is from sort of 2008. There's also sort of risk factors. So of course, a risk factor for skin cancer is sun exposure. A risk factor for breast cancer is actually your alcohol and how much alcohol you consume. Smoking is a risk factor. Actually, smoking is a risk factor, if you'll notice, for a lot of these. So by not smoking, you greatly reduce your risk of cancer. And you can see, if we look at the data, the screenable cancer that actually kills the most people is colorectal cancer here. If we catch colorectal cancer early and screen for it properly, it greatly reduces your chance of dying from it. So definitely risk factors are something to consider in terms of cancer. It's not just genetics. So in terms of sort of like study guide type purposes, we talked about operons. We talked about sort of gene expression, how selective gene expression allows us to differentiate. We talked about how DNA is packaged into our chromosomes, sort of wrapped around those little histone proteins. We talked about sort of that tortoise shell pattern, sort of that orange, slightly black pattern, because it's on the X chromosome. We talked about homeotic. Did I skip one? Yeah, I did. We talked about gene expression in eukaryotes. We talked about DNA splicing and RNA splicing. breakdown of mRNA, sort of translation and protein activation and protein breakdown, how sort of all the different ways we can sort of activate and sort of change our gene expression even if we have transcripts being made. We talked about microRNA, homeotic genes, and sort of how they play a role in development. We talked about a couple techniques for looking at gene expression, so things like the microarray and the nucleic acid hybridization. We talked about single transduction pathways, how it's sort of like a relay race that can trigger responses in target cells. We talked about cloning and sort of how differentiation affects or doesn't affect cloning. We talked about therapeutic cloning and sort of reproductive cloning, and how sometimes there are ethical guidelines there. We talked about oncogenes and tumor suppressor genes, and then sort of how cancers sort of snowball, that they may start off small, but then sort of snowball into these sort of unchecked, unregulated growth and malignancy in tumors. And then we talked about sometimes how the transduction pathways can screw up, which can lead to the development of cancer. And then we also talked about some risk factors for cancer as well. And that is the end of chapter 11.